with keen assistance from vet labs and extension services, farmers are likely to curb the major challenges that affect their herds.
Sentinel has taken 5 years, and if an animal is detected with the Rift Valley fever, so that's an indication that from this place they have now to go to send that to other farmers, so that they know that in Rift Valley we have the outbreak of Rift Valley fever. So it has helped us a lot during the rain season, because Rift Valley fever occurs mostly during the rain season. So during April, May, June, when we have heavy rainfall, that's when the spread of the mosquitoes, they spread very fast and they transmit that disease.
From the Sentinel herd, experts from RVIL are sent to collect samples to be taken to the lab for screening.
We have a team that is comprised of a veterinarian, 2 technologists, and a number of animal handlers. And we basically just get hold of the animals and using our tubes do the collection.
Serum is an amber-colored protein-rich liquid which separates out when blood coagulates. The blood serum of an animal provides immunity to a pathogen or toxin by vaccination or as a diagnostic agent. Back in the laboratories at RVIL, they ensure to test the serum to check on pathogens and agents that are present in the liquid, distinguishing the disease of an animal. Dr. Job Mungari, a technologist from RVIL serology section, takes us through the serum tests and what they survey.
In serology section, we normally deal with serum and milk. We test serum and milk for the presence of antibodies. These are reactions which come after disease. So after you have a particular infection or an animal gets an infection, it develops antibodies. So we test the presence or absence of those antibodies in the animal blood or milk. So our samples we normally use in serology for serological analysis are milk and blood, of which we take serum from the blood, which is clotted blood. What are the tests that we do here? The tests that we do in serology are like leptospirosis, brucellosis, and Rift Valley fever, among others. The serological test that we normally do to detect those diseases are agglutination test, that is latex agglutination test, and confirmatory test, which is ELISA. So we use ELISA test for confirmation of those and the presence of those antibodies in serum or in milk. When we are screening for the zoonotic diseases, brucellosis is among the zoonotic diseases, we use milk or serum. In case of milk, we do a test called milk ring test, where we detect the presence or absence of Brucella antibodies in milk. And when we put the milk and the Brucella antigen, we incubate, there is a kind of reaction which will be shown on the sample. For the positive sample, for example, they'll form a formation of a blue ring on top of a clear milk zone. For a negative reaction, there will be an homogeneous solution, meaning that the antibodies were absent. The Brucella antibodies were absent. So this is an indication that the Brucella antibodies are present in the sample. So apart from milk ring test, we have another test to confirm Brucella, which is called Ross-Pengo plate test. Ross-Pengo is a test which is done using serum, whereby we get our positive control and negative control. We normally run a test against controls, and then we put a particular amount of the serum with antigen, and then we rock for some few minutes, 4 minutes. We observe for agglutination. Presence of agglutination means it's a positive reaction, and no agglutination is a negative reaction. As you can see, there is some formation of crumbs. Those are the agglutinins. And the one which is a negative case, there is no agglutination. It's clear. This is a positive control, this is a negative control, and then this sample was positive, meaning that it resembled with the positive control, and the other sample was negative. Yeah? That is how we detect the presence or absence using agglutination.
From Top Farm, vet officers and technologists from RVIL inject the animals from the nerve at the neck region, where they collect the blood for serum extraction for screening.
Sentinel herds require special conditions. For example, these sheep that you see here are not vaccinated against Rift Valley fever. These sheep are also not, we don't do any control using acaricides or mosquito repellent on these animals. And the reason is because Rift Valley fever virus is a vector-borne disease. It's borne by the mosquito. So if we did control mosquitoes, we would actually be beating the purpose. We need to detect whether the mosquitoes are infected by the virus, and so these animals are not controlled. That kind of control of ectoparasite is not done on them.
After the lab test back at RVIL, lab technicians compile a report from the sample collected and share with the county veterinary offices that dispatch the information to farmers and advise best on how to prevent and control the livestock diseases before spread.
Information is cascaded down from the Central Veterinary Lab in Kabete to the district, now currently called the county veterinary offices, and then alerts are given in terms of what we call quarantine, so that animal movements are stopped until the disease is contained. Measures like vaccination for disease containment are also carried out.
For farmers to get candid and verified information from the lab, the process takes 2 weeks. This is to ensure that the results released are well researched and provide vivid confirmation from RVIL. At times, when the tests are impossible to predict, the RVIL lab offices send their samples to the Central Vet Lab in Kabete for further screening and confirmation.
It takes just 2 weeks. Just from this farm, they go to the laboratories, and then from the laboratories we get the feedback. So we know this animal has been detected. It has Rift Valley fever.
Having a Sentinel that assists farmers shows a great concern to the growth of livestock keeping for farmers in Kenya. Mr. John Kayere shares nuggets of wisdom involving extension officers in your livestock keeping as a business.
This is a national station, and so they do wait for our reports. So we sensitize them through veterinary officers in this country, so that they know there is an outbreak of Rift Valley fever.
The Sentinel herds should be visited at regular intervals. Ideally, animals should be sampled at the beginning of the rainy season and thereafter every 4 to 6 weeks up to the end of the rainy season. In a typical year, this will involve 4 or 5 visits to each herd. At each visit, some basic information is obtained and blood is collected from all the Sentinel animals. Samples should be forwarded to the Rift Valley Investigation Lab offices in Nakuru, Karatina, Garissa, Kapenguria or Kericho and tested within 2 days of arrival for the presence of antibodies. Therefore, farmers should take sheep and goat farming seriously by ensuring their herd are well protected from diseases that might wipe out a flock. They should also involve veterinary officers in their farms to assist in treating and vaccinating their herds against diseases. For more farming tips and knowledge on agriculture, keep it KTN Farmers TV.